Welcome to the CIBM Breakfast Science Seminar 3. It is my pleasure to introduce our second speaker, Tomas Ros, who is a research staff scientist at the CIBM EEG HUG-UNIGE section, and he will be giving a talk on neuroplastic effects of EEG neurofeedback.
So good morning, everybody. Thanks for coming. I'm going to talk about EEG neurofeedback. And for those of you who are not familiar with it or maybe experts with EEG, I'll provide first a big picture kind of point of view about EEG as well as neurofeedback and how they can be combined together, followed by some proof of concept studies using different modalities and techniques to validate neurofeedback and basically probe for the effects on plasticity, including EEG, TMS, and fMRI. So in terms of the big picture, I just want to start with this slide showing like the full continuum of where, we are in different zooming in scales of the brain, from starting from, the smallest levels of synapses and where medication works, and then going up more higher level to brain networks and circuits. So basically the traditional treatments that have been used for brain disorders have been medication, and now the new sort of generation of treatments like brain stimulation, like RTMS, the transcranial magnetic stimulation, as well as DBS and so on. They work more on the network level, so it's more higher level up, large-scale networks where sort of the closest to behavior you can get in terms of the overall distributed activity of the brain that we can record non-invasively. So neurofeedback works on this kind of network oscillation level where we can record things with EEG, and the beauty of EEG as well as the advantage is that it's non-invasive as well as sort of has a high temporal resolution, so you get a kind of real-time window of unfolding brain activity and how that may be related to behavior. But just starting with some basics of the EEG, I don't know, for those who are not experts, just one slide to sort of explain a little bit the dimensions that we use and we look at on EEG that's ongoing, so that's kind of spontaneous EEG, it's dynamic, it's constantly fluctuating, and this is the signal we use for neurofeedback, which is a kind of real-time method. And so the two main dimensions that we use is the amplitude of the signal and the frequency of the signal, which is kind of the Y and the X axis on this on the traces. And then we record these traces on multiple channels, and you can see the raw EEG is kind of a mixture of many different components, a little bit like music, has higher frequencies, lower frequencies, and they're changing in amplitude and time. And you can imagine it a little bit like the music on your hi-fi. You can kind of promote different components of the signal. So like slow frequencies like delta, theta, or higher frequencies like gamma and beta, and so on. So it's a little bit like the EEG is changing all the time, a little bit like music when you're listening. So different components are getting expressed at different times. And why is that interesting? Well, if you sort of throw away time and just take an average over a resting state of the spontaneous EEG, you can find that, in fact, in many brain disorders, including psychiatric disorders, there are differences compared to healthy controls, which indicates that maybe there's some kind of abnormality there that may be related to cognitive changes. So, for example, here there's an overall change in the controls versus patients. So patients have more power, that means amplitude of the signal in the low frequencies as well as the high frequencies on average. But then if you look at the individual traces, this is averaged over all electrodes. You can see some kind of disease-specific fingerprints that are different between disorders. And on top of that, you might also have spatial differences. So location of the generators may be different. So for example, in obsessive compulsive disorder, theta power is usually maximal in the medial prefrontal cortex, whereas usually in healthy controls, that's the case. It's more posterior, so that's more in the occipital regions. So these are kind of potential targets for treatments. See also, is there a kind of a causality there between changing the brainwave patterns and maybe changes in behavior? And there are different ways, obviously, of modifying these oscillations. One is you can use medication. You can also use other types of brain stimulation that target, for example, directly these oscillations, like transcranial alternating current stimulation, or as now the emergent sort of method is neurofeedback. So, but to be able to control these oscillations, I just want to start with a sort of neuroengineering perspective on this, which is how do you actually manipulate these oscillations from the point of view of neurofeedback. So the concept of control is obviously you're probably familiar with it in terms of a closed loop system. And the closed loop system has three components, which is sensing, computation, and feedback. And just to give you an example in terms of a thermostat, that's like the simplest closed loop system you can use. Is, for example, if you wanted to maintain the temperature of the room at 20 Celsius. So that's your kind of set point. And let's say that the sensor in the room found out that your temperature was 15 degrees, that means lower than the set point. So what happens is basically there's an internal mechanism that calculates the difference between the real-time temperature and the reference value, the set point, which is now -5. And then based on that, that will kick in, will engage the central heating units to raise the temperature, to raise sort of the temperature to start heating until you get to zero difference between the set point and the real temperature. And then the heating will switch off. So this is continuously happening. And if you think about the same principle in a cruise control, you have the same thing for controlling speed in your car. You sort of set a speed, and then the computer will calculate the speed currently, and then it will try to change the engine power to accommodate the speed to that speed. And what's interesting from this point of view is that, in fact, control is only successful for variables that are observable. So if you want to have best control of a particular variable like speed or temperature, the best is to have actually observe directly that variable. So have a sensor that observes directly the speed or the temperature. Otherwise, you might have some internal models that try to sort of approximate how these things are correlated. But because there's always external perturbances in the system, the best way is to control with a closed loop and have a direct measuring of that variable. Then the other thing is that this thing that's important is that you have high temporal accuracy, so that the thing can sample the signal in almost real time, hopefully, as well as that the accuracy is good. So this is what sort of is one of the principles behind neurofeedback, is that it provides, it enables unobservable brain activity to be observable. So in some sense, this is how we like to look at it, that you might know that you're in an attentive state, or you might know behaviorally that there is some kind of relationship between what is going on in your brain and the brain signals, but the neurofeedback enables a more accurate representation or a more accurate recording of what's going on in your brain at the same time. And that's essentially what neurofeedback does. It's kind of a mirror for the brain activity. And then you sort of look at that activity yourself and try to find a correlation between what you're doing cognitively and what is happening on the screen, and find a relationship between those two. So how does that work? Essentially, this is what the loop I described to you. So there's an element of sensing. So you take a particular parameter of brain activity. This can be, alpha amplitude or any kind of signal from the EEG. It could even be a connectivity signal. We can also do this with fMRI. And then you extract that feature, and then you classify it. So it can either be a continuous variable up and down, or it could be also like a thresholded variable where you say, okay, based on the resting state, this signal is higher than the mean or below the mean. And then you use that variable to basically re-represent it as a kind of visual display. It can also be auditory, but essentially you can display it as a visual signal. And then, for example, you have a spaceship that's moving, and that's related to sort of the fluctuation of that signal. How does that, so there's this computation aspect, and then finally the feedback aspect. So it's kind of a little bit like the closed loop that I showed you before. And how does that look in practice? Well, here's a replay of a neurofeedback session. Here, the central spaceship is linked to the alpha amplitude, and you have some other spaceships here that are also involved in artifacting. For example, if the person's clenching the jaw muscles or anything like that, then with a back signal, then you can use other feedback stimuli to give you a better signal, for example. But essentially, if you look at the middle spaceship, you can see how the blue bar graph fluctuates, and then when the blue bar graph goes below the middle line, the spaceship stops. So now it's moving, and then eventually it will fluctuate, and then will come back down to the bottom, and then it might stop. You see there the flame stops when the signal is below the sort of median threshold. So essentially, the person is looking at the feedback of this of the signal and tries to maximize or minimize that parameter over, let's say, a 30-minute session. So it's kind of a brain training, and then usually what happens is that they repeat this on a sort of daily basis or once or 2 times a week as a sort of plasticity of the training. So we'll get to that later. But just to show you how this looks like in terms of control. So here's one beta signal that people have been trained to modulate. And so you can think of it about sort of signal to noise training. So the noise will be the resting, the spontaneous activity. These are the white blocks. And you can see that the mean of those white blocks is lower than the training blocks. So the person sort of is in a spontaneous EEG state, then we ask them to promote this activity. They rise during the training blocks, and then but usually between the rest blocks, these things go down. So you could say, well, that's quite fancy. You can control brain noise. We know that people can control anyway brain activity. But why is that interesting from a sort of therapeutic point of view, and also maybe looking at the relationship between brain and behavior? Well, essentially, if you can change these signals more long-term, then we might find some kind of plastic changes also in behavior. And that's sort of the studies I'll show you here, proof of concept, to see how you can utilize this method to investigate these relationships and maybe find a new way of treating brain disorders. But essentially, the question we start with is that, is the brain just like a computer? And, you know, there's been a lot of these sort of parallels. But in fact, I think it's more like wetware rather than software or hardware. It means that the software changes the hardware and vice versa. So in some ways, by modulating brain function, you can maybe induce some plasticity. So this is some of the studies that have been used to study neurofeedback-induced plasticity. So starting with the EEG signal itself, what happens when you train this EEG signal for multiple sessions? This is one of the studies that show that if you look here, this is in healthy subjects. There's session 1, session 2, session 3, session 4, session 5. These are individual different neurofeedback sessions that are done on different days. And then within each session, these are the little small blocks that you can see that are like about 5 minutes each, that essentially where the signal is increased. And one thing to note here is that, in fact, the resting state signal at the end of the session is higher than the beginning. So there's kind of like short-term plasticity going on here. But also that if you look over time, over sessions, these kind of resting state increases gradually. It's not completely like along the regression line because there's some noise in the system. But overall, there is some, let's say, plasticity in the direction of training. And you can see that by the trace before and after, where at the beginning, in many subjects, the trace looks like that. It's kind of desynchronized, low amplitude alpha, and then you see this emergence of this alpha rhythm as people learn to promote this rhythm and generate it. So essentially, this is kind of a Hebbian plasticity model where you train in the direction and you promote more of the thing that you're training. Then, for example, one clinical application would be in brain disorders like autism or ADHD. There's quite a few studies on this already in neurofeedback, but this graph will show you that there's no, for those that decrease, so here the protocol was to decrease theta power. I showed you in the earlier slide that in some cases the theta power can be larger than the control subjects. So especially in autism, there's quite a lot of theta power in many children. So here, this bottom half of this children would be considered as responders, where you can see over many sessions, almost 40 sessions. So it's almost like going to the gym every day. You have some plasticity going on where this theta power decreases. This is the resting state power. So it's what happens after you train. So it's kind of impressed into the system. And although you can also see that there are some children that do not respond to the treatment, i.e. they do not have any kind of plasticity, and the sort of baseline power remains the same over sessions. So those are two examples of long-term plasticity of the EEG oscillations themselves over time. But getting to sort of using more robust evidence of any other kind of changes that we can say to support those changes in the EEG. And this is where we started to do some studies where we wanted to look at, okay, are there any other windows on the brain that we can use to sort of show that you have some changes in the EEG in the brain, and as well as maybe look for changes that are within a single session. So here there's quite a lot of sessions, but could you actually see a change between each session? Is that even tangible? Can you measure it even? Which is what you find also with some other types of brain stimulation. So we collaborated with Queen Square in London, the Institute of Neurology, the Rothwell Lab, and we tried to use magnetic brain stimulation to look for changes in plasticity. And I don't know if you guys are familiar with TMS, but essentially what happens is you stimulate part of the motor cortex, and then you look at the evoked response on the muscle, which is essentially proportional to the degree of connectivity, synaptic connectivity between the motor cortex and the muscle. It's a trisynaptic pathway, and essentially the more connectivity or the more synaptic strength there is between the motor cortex and the muscle, the bigger this response that you have, this motor evoked potential, the MEP, the motor evoked potential increases as synaptic potentiation increases, or excitability of the motor cortex increases. So what you can use this kind of method to look at, okay, are there any short-term changes in this pathway pre to post treatment, for example, neurofeedback. So, and also to choose a neurofeedback protocol, what kind of brainwave should we modulate? There are some relationships there between metabolic activation and the EEG. For example, here you can see these low frequency waves in cortical sensory areas are negatively correlated with metabolism. So in the fMRI, for example, this bold signal increases when these alpha and beta and theta rhythms decrease in general. And for the faster rhythms, the high frequency rhythms is the inverse. There's a positive correlation. So essentially, the reduction of alpha of the sensory sort of dominant rhythm is related to an increase in bold. And we also know from previous simultaneous EEG TMS studies that if you stimulate with TMS at particular phases or particular parts of the EEG when it's fluctuating randomly, that you will have a higher MEP here, that's the blue, when the alpha is low. You can see here the EEG band amplitude. When the alpha amplitude is low, then you have a high MEP is generated, and when the alpha amplitude is high, you have a low MEP generated. In some sense, there's this inverse correlation between alpha and the excitation of the cortex, or the motor cortex. And this is in line with the sort of theory that alpha acts as a gate for a sensory processing. So essentially, higher alpha means the gate is closed, lower alpha means the gate is open. Okay, and so you can use that to say, investigate, okay, is there a neuroplasticity effect of training the alpha rhythm? So what we did was we tried to look at, after a single session of neurofeedback, which is here, of reducing alpha, what happens to these MEPs? So we take a baseline MEP measurements on the left motor cortex as well as the right motor cortex, and then we look at what happens to the MEPs post neurofeedback, and do they sort of increase or decrease due to this one single session. And as I showed you before, as I mentioned here, we do a single session, a left motor cortex, we had 12 subjects, and we measure these MEPs, and then we decrease this alpha rhythm at C3, which is left motor cortex. Okay, so the hypothesis initially would be that since you're decreasing alpha, you would find an increase in excitability, right, because of this inverse relation. So that's what we hypothesized. And what we found, firstly, for the training EEG, is that compared to the resting state alpha, which is block 1, each block here is 3 minutes. So we train for 30 minutes in total. You can see that the resting state alpha is much higher than the training alpha periods. And you can see that they kind of gradually decrease in time over on average for the whole group. So that means that the neurofeedback training was successful. People can like imagine things that they're doing with their hands that decrease the alpha rhythm usually. They're also called the mu rhythm, or they could even pay attention, and that sometimes desynchronizes the EEG. So the actual sort of manipulation, it was successful. Then what happened to these TMS parameters? And you can see here at T1, this is immediately after neurofeedback. The left hemisphere, in fact, MEP didn't change. It's almost the same as baseline, 100%. But gradually over time, which is consistent with some kind of plasticity mechanism that engages over time, this MEP excitability increases. So the MEP size on average is almost 25 to 30% higher than the original. And in fact, it correlates with the time elapsed. So it's not an arousal effect, because an arousal effect would have maximal changes at the beginning and then it would drop with time. Here it's almost the opposite. So it gives us some confidence that maybe this is some kind of molecular mechanism going on that increases excitability or induces the plasticity in the system. And then obviously this is the group changes, but are there any individual changes that we can correlate with the training efficacy itself? And here we have a 3-way scatter plot that we observed essentially between the normalized changes during training and the rest state. So in fact, the more people decrease alpha during training, the more the resting state alpha decreases after training, okay? And that's correlated to the MEPs. So you have this 3-way relationship between training EEG, resting EEG, and changes in MEP, which is quite nice. And just note here that, for example, the training EEG is correlated to the MEP. So here and here you have this negative correlation, which is significant, which is important. But if you do a structural equation model, then in fact, that correlation now becomes insignificant. So in some ways, a potential mechanism or model for how neurofeedback works is that the training EEG induces a change in the spontaneous EEG post-neurofeedback, and it's the spontaneous EEG that mostly predicts the changes in MEP. So this is in line with the fact that this activity training impresses the same signature in the resting state, which then leads to this change in the excitability of the brain. And just to overlay this type of treatment or this type of manipulation with traditional ones that we already know about, like RTMS, which is repetitive TMS. Repetitive TMS, so TMS can be used to look at plasticity changes, but if you apply it repetitively, you can use it as a kind of neuromodulatory tool. So you can use it to also enhance excitability. Now, some other studies have shown that if you apply TMS in a very fast sort of train over at least 25 hertz, 20 hertz, you can also induce plasticity. So you see here 1 hertz, 10 hertz, 15 hertz, and at the beginning there's no change in excitability, but as you increase the frequency, you start to get increases in excitability due to this type of manipulation. And neurofeedback, according to the effect size as well as the error bars from this study, show that it does kind of match the RTMS sort of effect size. So it might be an interesting sort of method to use also to increase excitability and also to substitute in those areas where TMS is used as a sort of neuromodulatory tool in brain disorders. And just to show you also that this can be also interestingly can be looked at from a point of view of medication. Also the medication can change the TMS parameters. For example, methylphenidate, which is sort of an agonist of noradrenaline, usually increases MEPs, and diazepam decreases MEPs. So neurofeedback has also been used in ADHD, for example, where methylphenidate is the frontline treatment, and maybe the mechanisms are similar in the fact that it enhances excitability, at least for this protocol. So we don't know, but there's a lot of connections still to be made in this arena. And then the last type of method that we could use to look at neurofeedback effects is fMRI. Now, of course, there's also fMRI neurofeedback, but I'm talking about EEG neurofeedback here and using fMRI to look at changes in the brain. And again, we focused on studies using alpha. Essentially, alpha is very easy to control with neurofeedback. And there are some relationships between alpha and behavior. So we went ahead and looked at the relationship now with alpha and in the parietal and occipital areas, which is more linked to attention and visual attention. So there are studies showing that, of course, as I told you, that the gate, when the gate is high, then there's less sensory information coming in. So it's the same thing for visual detection. And you can consider that this alpha fluctuates. And when it's high, that these are kind of internally oriented states where you're thinking about things that so you're kind of decoupled from the environment. And then when the alpha is open or closed, I mean, when it's low, this sort of you're more aware of the environment in terms of sensory information. But that goes both for the visual areas as well as the motor areas. So you could think that your brain is constantly fluctuating, alternating between internally oriented and externally oriented states. And usually when you're thinking, the alpha is higher and you're kind of internally oriented state. And then when you're kind of paying attention, the brain is kind of silent, then you're externally oriented. So could you use neurofeedback to, let's say, enhance the prevalence of this externally oriented states by modulating alpha again, by decreasing it? And interestingly, what are the fMRI correlates of that? We know from simultaneous EEG fMRI studies that, in fact, there's this kind of positive relationship between alpha and the default mode network. The default mode network is a network that's been linked to self-referential processing as well as mind wandering. And there is a relationship between alpha and fMRI. So in some ways, one would predict maybe that if you're decreasing alpha, that you would sort of decrease this connectivity in the default mode network. And as we also know that default mode network is linked to mind wandering. Also, again, since alpha increases, you would be more internally oriented. So in some sense, decreasing alpha would decrease the amount of mind wandering. So those were the two hypotheses, that neurofeedback alpha down regulation would decrease connectivity in the default mode, sort of down regulate this fMRI network, which is linked to internal processing, as well as decreased mind wandering, okay, behaviorally. So there's the triad of behavior as well as fMRI and EEG. And so in this study, we try to reduce alpha at PZ, which is a parallel area where alpha is sometimes maximal. And in this case, we also had a sham neurofeedback group. So essentially here, we replay a fake EEG signal from, in fact, a subject that it's not actually fake. It's pre-recorded from a previous session of subjects. So it's kind of ideally matching somebody who does well at neurofeedback, but because it's a replay, it has no real-time relationship to the person's EEG. So it's kind of a mock feedback, if you wish. And we just play that to people and say, okay, can you try to change this activity without telling them that it's actually fake? So it's a control for both the cognitive aspects of trying to find a relationship between the signal and the actual signal itself. And a similar design to what we used in the TMS study, we have a sort of baseline period where we do the different type of modality, so fMRI in this case. And during the fMRI, we have an intention task, which actually samples for mind wandering. You can look at the paper to see how we did that. Then we take the person out of the scanner, we do an EEG resting state, and then we do 30 minutes of neurofeedback, followed by another EEG resting state, and then go back in the scanner and do the attention task with the fMRI again. And we look at the fMRI networks, if they're modulated after neurofeedback. And so what did we find between these two groups in terms of the alpha training amplitude? Well, you can see here 15 is kind of the average for both groups in the resting state. And as we saw also in the TMS study, that people can modulate the alpha rhythm quite well to decrease it, to sort of decrease this gait. And then they keep this very low for almost 30 minutes during the neurofeedback session, and the alpha goes back up during rest afterwards. And what's interesting is that the sham group does decrease it because they're paying attention to the screen visually. So you would expect a little bit of alpha desynchronization at the beginning, but with time, this alpha goes back up because there's no relationship between their brain states and the signal they're receiving. And you can see that by this differential between the correlations. So there's a difference between groups in terms of the overall alpha amplitude, as well as a difference in correlations. So we would then expect maybe some differences in the fMRI correlates. This is just a topography. We did a full cap EEG during the neurofeedback, and just to show that you have this alpha minimum in the parietal areas as expected, which isn't so much the case for the sham group. There are also some changes in the other bands, but these were not sort of part of the hypotheses. But you can see here, beta has a slightly different topography. So there's again some difference between the groups there on the EEG. And the interesting thing that we found is that if we correlate the relationship between training on the x-axis of the alpha compared to the baseline and the resting state change post neurofeedback, again normalized values, there are differences between groups. So for the sham group, although there are some changes during training in alpha, those changes don't necessarily translate into some resting state changes, as you can see here on the y-axis. So there's no significant correlation between the two for any of the brain regions looked at, whereas we replicate this positive relationship that we found in the TMS study. So in fact, more alpha decreases during training, more it impresses in the resting state afterwards. That's almost like a global effect. Right? So it seems like it's a kind of global neuromodulatory effect, probably thalamocortical. So that's interesting. And then what were the relationship with these changes with the behavior? Well, we sort of confirmed this hypothesis that more alpha in the resting state after neurofeedback is related to less mind wandering. Less alpha during after neurofeedback and less mind wandering. So that's in line with sort of the brain behavior sort of studies beforehand. And then for the sham group, that wasn't the case. So in some ways, you could think that neurofeedback could modulate behavior in those people who respond to it. And in terms of improving attention or decreasing mind wandering, might be an interesting option. And then in terms of the
sorry, can you just go back?
Yes.
It just looks like the vast majority actually have the opposite effect.
Yeah. So in terms of the group change, there are some people who increased alpha, and in that case, they basically didn't decrease mind wandering, but increased it, and that was one of the limitations of the study. That in fact, at baseline, some of the subjects didn't have any mind wandering to begin with, So we had a sort of ceiling floor effect in terms of mind wandering. And so those people went in the opposite direction. But as I said, for those who responded in terms of impressing the alpha amplitude after neurofeedback, those do decrease mind wandering. So it's not necessarily a study that shows that neurofeedback per se, as a whole group, can improve mind wandering, but it does show that some individual differences. And some of these individual differences could be explained by the fact that they had zero mind wandering at the beginning, which means that the system is kind of optimal not to mind wander because they're paying attention. But for example, those subjects that have high wandering to begin with, or even subclinical, clinical cases, might be useful. So that's still something that we need to investigate.
Okay, thank you.
Yeah. And for default mode, these again, the brain behavior relationships, in terms of mind wandering default mode, we did replicate this relationship, positive relationship between these kind of default mind areas like precuneus and increase in mind wandering, or decrease vice versa. So less connectivity in the precuneus with this ICA network predicts less mind wandering, or vice versa, more mind wandering if it increases. And the same thing for the resting state alpha individually. So there's a relationship again with this alpha change and the mind wandering change that you can see on the MRI maps. And lastly, with what we did find in the group change as a whole between the two groups is that there was this increase in salience network. So in fact, the salience network, which is a kind of positive cognitive control network, we found an increase in connectivity in these middle cingulate regions that wasn't the case for the SHAB. So there was some kind of differential change on the brain, although there wasn't one that we detected in behavior as a whole. And this is consistent with this study here that showed this antagonistic relationship between this cognitive control network, especially in this dorsal ACC node, that is antagonistic to the default mode. So again, here you can imagine that these two networks are competing with each other between internal external processing. So let's say this red network would be more external processing, and then when it flips to internal processing, then it's the default mode that takes over. So that's in line with our results. So that's my last slide, and thank you for your attention, and also thank you to the collaborators that we worked with. The last study, the fMRI study, was done in University of Western Ontario with a group of Ruth Lanius, and we're now applying this maybe to some psychiatric disorders. We'll have to see what the results are, and of course also the UCL at the Queen Square in London. So thanks, and I'll take any questions.
Thank you.
Okay, thank you. Yes, any questions? We are way over time, but I think I underestimated the time anyway. So, anyone?
I might have some stupid questions.
Mm-hmm.
You started with acoustic stimulation and EEG measurement, right? You started with acoustic stimulation and EEG measurement, right?
You're talking about which study?
When you started at the beginning, your first
Yeah,
slide.
I was using music as a metaphor, the acoustic aspect, in terms of frequencies, but that's what you mean?
Yeah. So I have the following question. Of course, if you use the standard fMRI, you cannot use acoustic stimulation, right? But
we use headphones during fMRI.
Are they this good?
Yes,
that
you can do auditory experiments in the fMRI. So, for example, here for mind wandering, so what happened during this attention task? You can look at the paper in your image, but we had a thought sampling. It's like an audible task where you have some stimuli that are short and long. So it's like beep, 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 and the people have to detect the long beep in a sequence of short beeps, and then that's the low attention task. And then randomly interspersed within this task, there are some telephone rings that ask the person, are you on task or are you off task? That means, are you thinking about something else, or is it that you're completely paying attention to the task? So we rely on their honest evidence, obviously, of their thoughts to get a measure of the mind wandering. We also had a correlation with the reaction time. So these correlations here that you see between mind wandering and resting alpha are actually the thought sampling probes, the telephone rings, the change in the number of times they declared that they were mind wandering, but there's also a positive correlation with the reaction time. So you could say that maybe during those times where they had less alpha, or less alpha was induced post-neurofeedback, that they were more reactive, more attentive, more alert, let's say, to the telephone rings as well as the reaction time. Right? Does that answer your question, or?
Yes, thank you.
Okay, thank you.
Any other questions?
Yes, I have a small question.
Mm-hmm.
Hi, thank you for this talk. It's really interesting.
Thank you.
I have a question about this picture that you showed in the beginning with the spaceship.
Mm-hmm. Mm-hmm.
So did I understand correctly that so in the middle is the alpha rhythm that
Yes.
people try to control, and on the left side are people trying to control kind of the noise level?
Yes, it's sort of well, in the EEG there are some frequencies that overlap with sort of non-cerebral activity, like muscle activity.
Right.
So
Yes.
in the muscles you can generate some beta.
Yeah.
So, this right sort of spaceship is actually the beta activity.
Oh,
so
I
it's
see.
higher than alpha. So if they start to really get nervous and start to make facial grimaces or whatever, then this will go up, and then that will tell them, okay, relax.
And how well does that work? I'm just curious about this, because I mean, it could improve signal to noise ratio in many different settings, right?
Yes. Well, it works reasonably well if you're not training the beta band. So it's
Right.
effects are not orthogonal to the actual brain activity. If you're training alpha, it's okay because it starts with a higher amount, and then that can bleed into the alpha. But usually this will be higher. So it works reasonably well. And now there's some automated neurofeedback units can use AI to give you a sort of signal quality index.
Okay.
So it can help with children, for example, that don't know how to begin to train, and tell them to relax. But of course, it's all about how reliable the signal is as well. And you might be able to place separate electrodes really directly on the muscles to improve it, rather than capture the. So there's different ways of going about it.
Okay, interesting. Thank you so much.
Thanks.
Okay. Any other questions? Okay, if there are no other questions, I would like to ask you if you are okay with taking a group picture